Okay, welcome back. So we are going to go ahead and start the material, the last bit of material for 201, which will be the nervous system. And of course, we save the best for last. So the nervous system is very complex, but also really, really interesting. And we're just going to be scratching the surface in terms of the nervous system. And, there's so much that isn't known about the nervous system still. So, this is just a little bit of the information that we do know. So we're going to go ahead and talk about the brain and the cranial nerves in this chapter. And you'll see there is quite a bit to that. And then we'll get into the rest of the central nervous system, which will include the spinal cord. So when we talk about the brain, again, the brain is this center that's going to register all of our sensations, right? It's going to take that information in. It's going to, again, make associations with that information on what to do, make decisions about what to do, correlate that information and store it. And then also, again, as it's making those decisions, it's going to determine what type of action to take and send, again, that information out through the motor neurons. So our brain is also our center for, as you probably well know, intellect, emotions, behavior, and memory. It's really, again, what makes us who we are. So when we talk about the central nervous system, or the CNS, is going to be composed of the brain and the spinal cord. You probably already know that. Now, when we talk about, again, human anatomy and physiology, we are going to use the word cephalization. And what that means is this is just an elaboration of the anterior portion of the central nervous system, which is, again, our brain. And if you look at kind of evolutionary history, right, the cephalization and the development of cephalization, and as the progression, I should say, of cephalization, really is associated with organisms that have these abilities to do higher thought processes. And so when we talk about humans have achieved this kind of highest level of cephalization. And so again, as we again have this greater cephalization, we increase the number of neurons in the head. And you'll see this in terms of like the development. And we're not going to get into the embryonic development of the nervous system because honestly, you could have a whole chapter on that, and we just don't have time in this course. There's actually a whole course called embryology, which is really interesting if you ever get the chance to take it. And it goes through kind of, all the different, a lot of different vertebrates, the embryology of different vertebrates and how that progressed. So it's interesting course. But you can see that we kind of start with this tissue that forms what's called a neural tube. And that neural tube is what's going to become our central nervous system. And so you can see that that neural tube in humans is, again, going to have this progression of cephalization, where we really have this, an anterior portion of the central nervous system that is going to, again, be enlarged, more elaborate, okay? And that is ultimately what's going to become the brain. So that's going to eventually become the cerebrum and what we call the diencephalon, which we're going to talk about in just a minute. And then the brain stem, some of that, again, the brain stem will be the midbrain pons in the medulla oblongata. And so again, when we talk about the adult brain, okay, and no, you don't need to know these different embryological terms and what they become. This is just to kind of show you that progression of cephalization. Now, in an adult brain, we have four major regions. We're going to have the cerebral hemispheres, or the cerebrum. And this is really, again, what you kind of think of in terms of, most people think of in terms of the brain, where a lot of your higher thought processes and decision-making is going to occur. And then we're going to have this portion of the brain that's called the diencephalon. And the diencephalon will be broken up into the thalamus, hypothalamus, and epithalamus. And we'll talk about this kind of being a big relay station, but also plays a big part in the endocrine system, which is the hypothalamus. And then we have the brainstem That's going to consist of the midbrain, the pons, and the medulla oblongata. And then we're going to have the cerebellum and the very posterior portion of the brain there. So let me go through these different parts again. We have the cerebrum, and the cerebrum is broken up into the two cerebral hemispheres, so we'll talk about in just a minute, by this longitudinal fissure that goes down the middle. And then we have the diencephalon here. Again, we'll talk about that being made up of the thalamus, the hypothalamus, and epithalamus. And then we have, again, the brain stem, which is going to consist of the midbrain, then the pons, and then the medulla oblongata here, kind of going over this to help you with lab as well. And then you have the cerebellum back here, okay? So just a few reminders in terms of definitions as we were talking about the brain. Remember that when we talk about a cluster of neuronal cell bodies in the central nervous system, we're going to call that a nucleus. And when we talk about a bundle of axons in the central nervous system, we're going to call it a tract. So when we're focused on the central nervous system here, we're going to be using the terms nucleus and tract. And I just wanted to remind you of what those terms were. I'm not going to use the term nerve when I'm talking about a bunch of axons, a bundle of axons in the central nervous system, again, in the brain or even the spinal cord, because it's called a tract. Now remember that white matter is myelinated axons. So again, they're going to be covered with that myelin sheath. And then gray matter is going to be some of our unmyelinated axons, as well as anything that basically isn't an axon, right? Cell bodies, dendrites, all of your neuroglia is going to make up your gray matter. Okay. So when we look at the different brain regions, again, we're going to have gray matter and white matter. And again, remember that white matter is going to consist of myelinated, and then it will have some non-myelinated axons in it. Even though, again, the majority of the non-myelinated neurons are gray matter, but sometimes, again, in the mix of that white matter, you can have non-myelinated axons as well. But mostly, it's myelinated axons, and that's why it's white. And you're going to see that there's going to be a basic pattern found in the central nervous system. And that is that we're going to have the central cavity that we're going to talk about. That's going to have cerebral spinal fluid inside of it. And then that central cavity will be surrounded by gray matter with white matter to the exterior of that. Okay. So I'll show you this pattern here in just a minute. And this pattern will change a little bit between the spinal cord and the brain. So you'll see the spinal cord is really going to exhibit this basic pattern, although this pattern is going to change as we ascend into the brain, especially through the brain stem and up into the brain. And you'll see we still kind of have this basic pattern, but we're going to also end up having this kind of like bark-like layer around the white matter. That will be a little bit of a gray matter layer in the brain. So this pattern will change a little bit. But again, for the most part, you usually have, again, the central cavity, some gray matter around that, and then some white matter around that. So as we look at a cross-section of the spinal cord here, you can see that it exhibits this typical pattern. You have the central canal right in the center, where the cerebral spinal fluid is going to run through. And then you've got this gray matter that's going to surround that central canal, and then the white matter surrounding that. Okay. So as we move farther up, my slide will advance, there we go. As we move farther up into the brain stem here, you're going to see that it's going to change a little bit in that the brain stem is going to have these kind of additional, so it still has, again, that inner gray matter surrounding the central cavity, but now we're going to have these kind of additional islands of gray matter that are kind of buried within the white matter, and we're going to call these little islands of gray matter brain nuclei. So these are going to be called, those are the nuclei, okay? And again, they're going to be kind of buried in this outer white matter layer. So we still have that kind of basic pattern that you can see with the white matter on the outside. It just has these islands of gray matter called the nuclei. Now, as we move again farther up here into the cerebrum, we're going to see, and you'll see this pattern in the cerebellum as well. You're going to see that we still have these central cavities, but now the gray matter that was directly kind of surrounding that central cavity has now kind of been left as these little islands of nuclei. And then we have, again, a large area that's, again, the white matter, this large outer white matter, but now the very outer layer, okay, that we're going to call the cortex is going to be gray matter. So the very outer layer here. And the reason that they call this the cortex is it actually gets its name. I put this down here to help remind you. It actually gets its name from the Latin word that means bark of a tree. So if you look, this gray matter actually forms kind of like this layer like the bark would on a tree. And so again, we have that kind of outer layer of gray matter. And remember that gray matter is going to be where we're going to find our neuron cell bodies, and again, our short non-myelinated neurons, and as well as our dendrites too. Okay, so that's just kind of the basic pattern distribution of what we're going to see in terms of gray matter, white matter throughout the central nervous system. Now we'll get more into that as we talk about the different parts of the brain. So again, we're going to kind of get more involved into that. Now let's talk about, we talked about the central canal and the spinal cord that holds the cerebral spinal fluid. But if you look, the central canal kind of enlarges here in the brain, and it forms these much larger kind of hollow cavities of the brain that we call the ventricles. And these ventricles of the brain are very important because they not only are these, again, fluid-filled cavities or chambers that are going to hold the cerebral spinal fluid, but they're also actually going to make it. In terms of, well, I shouldn't say they specifically make it, but the choroid plexus, which is going to be, and again, I'll have a slide on this in just a minute, but the choroid plexus is going to be a bunch of blood vessels that are going to be on top of, the ventricles here, and then even this third ventricle here, that are going to make that cerebral spinal fluid. So all of these blood vessels here, there'd be like a grouping of blood vessels up here, and then some of those fluids that are really most important for the brain and are most important for the functioning of the neurons will seep out of the blood vessels of that choroid plexus and into these hollow chambers that we call the ventricles. And then we'll talk about how that cerebral spinal flows, again, through here into the spinal cord as well as around the brain, because that cerebral spinal fluid will also bathe the brain. So when we look at these hollow cavities in the brain, again, that contain that CSF, the cerebral spinal fluid, keep in mind that these cavities are going to be lined with those ependymal cells. Remember those? We talked about those in the last lecture. And those ependymal cells are the neuroglial cells that are going to form that like brain cerebral spinal fluid barrier. And the ependymal cells are ultimately, going to be making that cerebral spinal fluid as they're filtering that fluid from the blood vessels, from the choroid plexus blood vessels into those chambers, okay? And so again, these chambers, just to kind of go through what the different chambers are, because you'll need to know these for lab for sure. You were going to have these two lateral ventricles here. And then these two lateral ventricles will be separated by something called the septum pellucidum. And then the two lateral ventricles will then connect to what's called the third ventricle here through, this is nice, the name is interventricular foramen, right? Because it's between the ventricles. So between the lateral ventricles and the third ventricle. So to drain into that interventricular foramen, into this third ventricle, which is actually in the diencephalon. And then from that third ventricle, then it will drain down the cerebral aqueduct into the fourth ventricle. Now in this fourth ventricle, you'll see it's got one opening out here into the central canal. But then it also has 3 other openings. So 1, 2 here you're going to see are the lateral apertures, and this is going to drain into what's called the subarachnoid space. This will make more sense in just a minute when I go over the different kind of meninges of the brain. But they're going to lead into the subarachnoid space, as well as this one here called the medium aperture. So those 3 openings, the median aperture and the 2 lateral apertures, are going to, again, leak the cerebral spinal fluid into the subarachnoid space, and that is going to bathe the entire brain in that cerebral spinal fluid. So pretty cool. So let's talk a little bit about what the cerebral spinal fluid is. So cerebral spinal fluid, we, again, have about 80 to 150 mils, or 3 to 5 ounces in our body at any point in time. Obviously, this is going to vary between individual to individual. And the cerebral spinal fluid should, in a normal individual, be absolutely clear. I have actually had the luxury of actually getting to see this firsthand when I did multiple sclerosis research. One of the things that we do is we look for certain inflammatory markers in the cerebral spinal fluid. And so I do a lumbar puncture and collect some of that cerebral spinal fluid. And it should look like this. It should be absolutely, completely clear. If it looks like this or like this, that's usually a really bad sign. Usually an individual in that case is not feeling well, because that is an infection that is in the cerebral spinal fluid. And that's the cerebral spinal fluid that bathes the brain. So obviously that can cause things like, that can be caused by things like meningitis. But typically, again, cerebral spinal fluid would be clear, and I mean crystal clear. It contains glucose, again, for the brain, because the brain, again, isn't making its own glucose, right? So it actually has that cerebral spinal fluid, again, as well as, remember, those astrocytes are going to help bring some glucose in from the blood vessels to those neurons. Again, those are cerebral spinal fluid is one way that we can bring nutrients to those neurons, as well as certain proteins and ions. And again, that cerebral spinal fluid is also going to create a condition that's ideal for the functioning of the neurons. So it's going to be the right pH, right? It's going to create that ideal condition for everything to function properly in the brain. So that cerebral spinal fluid is really important. So again, the functions of the cerebral spinal fluid are obviously, again, nutrients. It's going to provide nutrients, as well as it's also going to get rid of waste products from those neurons. Okay, and then we'll talk about the flow of cerebral spinal fluid in just a minute. Also, it's going to provide some mechanical protection. So it's going to act as like a shock absorber, right? So again, if you can picture the brain being kind of bathed in this nice liquid layer, right? But between the brain and the skull, that helps to kind of act as this nice shock absorber, so the brain's not bouncing right against, you know, the bones of the skull. So kind of important there. And then it's also, again, going to provide some of that chemical protection. So it's going to provide the ions that are needed for action potentials. So we just talked about how important sodium and potassium ions are, right? So again, the cerebral spinal fluid will provide those ions. Now, a reduction of cerebral spinal fluid turnover can really cause some issues, and it can cause the accumulation of certain metabolites, certain byproducts of metabolism, but because those neurons, again, are constantly metabolizing things like glucose to be able to do what they need to do, right? And so in aging and neurodegenerative diseases, we oftentimes see that there's a reduction in that cerebral spinal fluid turnover. So we're not properly getting rid of those waste products. And this is one of the kind of thoughts of Alzheimer's disease, is that, you know, when we sleep at night, a lot of that cerebral spinal fluid turnover is occurring while we're sleeping at night. And so those metabolites are actually, again, pulled out of that cerebral spinal fluid so that they, again, aren't clogging up the neurons in the brain while we're sleeping at night. Now, we talked a little bit about that choroid plexus. So the choroid plexus is just going to be this series of capillaries that are, again, going to be covered by the ependymal cells, right? And the ependymal cells are going to filter, again, the liquid from those capillaries of the choroid plexus. So all these blood vessels are going to be sitting, like there'll be a bunch of blood vessels right on top here of the lateral ventricles. And the ependymal cells are going to filter that fluid from the choroid plexus, and it'll bring that fluid that's going to make the cerebral spinal fluid into those lateral ventricles. And then you're also going to have some choroid plexus up here on that third ventricle. Again, it's going to be doing the same thing, as well as some even on the fourth ventricle. And so, again, those ependymal cells are going really to filter out the right things that are needed to make up that cerebral spinal fluid, and make that perfect, again, ideal condition for neuron functioning. So again, you're going to see here the lateral ventricles here are going to then drain into, as cerebral spinal fluid's made, the lateral ventricles will drain through the interventricular foramens into the third ventricle, and then from the third ventricle through the cerebral aqueduct into the fourth ventricle, okay? And then down again through that central canal. So we kind of talked about that. And then it's going to move out those, remember those three openings that we talked about, the paired lateral apertures, as well as the median aperture. So those were, again, we had the paired lateral runs on the sides here coming out, as well as that median aperture in the middle. And those are going to drain into what's called the subarachnoid space. Now, to explain what the subarachnoid space is, I have to explain what the meninges are. So the meninges are these three layers, okay, that are three kind of membrane layers that are going to surround the brain. So they'll be in between the brain and the cerebral spinal fluid. Well, kind of, the cerebral spinal fluid will be in between there. And so the three layers of meninges that we have are going to be the dura mater, the arachnoid mater, and the pia mater. Now, you're going to hear them pronounced both ways, dura mater, dura mater. They're both correct. It's just going to depend on individual to individual, as well as hospital to hospital on what you're going to hear. But again, it's one of those things like, you say tomato, I say tomato, same thing here. You say dura mater, I say dura mater. Both are correct. Whichever pronunciation you like, you can stick with. I'm going to use my traditional dura mater, arachnoid mater, and pia mater, just because when I took anatomy, that was how it was taught. So when, again, the cerebral spinal fluid leaks out through these lateral apertures in the median aperture into what's called the subarachnoid space. That subarachnoid space is going to be that space in between the pia mater and the arachnoid mater. Hence the name subarachnoid, below the arachnoid space. So when you look at the layers of the meninges, let's see if I have a better image of this. When you look at the layers of the meninge, you're going to see this outer layer is going to be the dura mater. It's going to be the toughest layer, and if you were doing a dissection in the lab, this would be the layer that's like really the tough layer to cut through. So you're going to have the dura mater here. And then you'll have the arachnoid mater right up against that dura mater. And that arachnoid mater it's going to have this kind of fibrous content to it. So because it's going to have these kind of fibers running through it that are kind of web-like, it's given the name arachnoid mater. Arachnoid, like, again, spider. So they thought it looked web-like. So you're going to have that arachnoid mater, and then you'll have the pia mater that's directly around the brain. So the pia mater will be this layer, this membrane that directly surrounds the brain. So the subarachnoid space is literally, just like it sounds, below the arachnoid mater, in between the pia mater and the arachnoid mater. And this subarachnoid space is where you're going to find that cerebral spinal fluid. So that cerebral spinal fluid, again, is going to move out the lateral apertures and the median aperture to travel into that subarachnoid space. And as it moves around that subarachnoid space, again, it will bathe the brain, okay, in that cerebral spinal fluid. Now that cerebral spinal fluid, again, is creating the ideal conditions for, again, the functioning of those neurons, as well as, again, it's bringing in, remember, ions and glucose and removing waste. Now, it's really important that it's bringing in that glucose because, again, your brain needs that glucose. It doesn't have its own storage content of nutrients. It doesn't have this like fat storage that it's like drawing from directly in the brain, right? So it really relies on that glucose in the blood to be brought in, again, through the cerebral spinal fluid, as well as through, again, the astrocytes directly as well. So let's talk about how, again, this flow of cerebral spinal fluid happens. So again, we're going to be making cerebral spinal fluid at the choroid plexus of each of these ventricles. So each of these ventricles has, again, the choroid, and here the choroid plexus, like when you look at your models and stuff, it's just going to be drawn in with like a bunch of like red web-like lines. That's the way that they're kind of showing you all of these capillaries, right? These blood capillaries that would be around that area. So the lateral ventricles are going to have choroid plexus on top of them. The third ventricle, which is what they're showing here, is going to have choroid plexus on top of it. And then the fourth ventricle is going to have choroid plexus on top of it as well. So they drew that in here. And again, all of those are going to be making that cerebral spinal fluid through those ependymal cells. So as that cerebral spinal fluid is made and enters again into each of these ventricles, so as it enters the lateral ventricles, it will move through again that interventricular foramen into the third ventricle here. And then from the third ventricle, it will move through the cerebral aqueduct, again, into that fourth ventricle, and then through the lateral and median apertures, okay, out into that subarachnoid space. And then, of course, some of it also will move down again into that central canal of the spinal cord as well. But as it moves around that subarachnoid space, okay, it's going to have to be drained off of there, right? And there's really two reasons for this. One, you would just build up fluid, cerebral spinal fluid around the brain, and that would be really bad if it wasn't ever drained off. But also, again, we want to drain off all of those metabolites and waste products, right? So as we do that, what's going to happen is you have these little things called arachnoid villi. They're right here, they're drawn in right here, these little kind of like flower-like structures here. These are the arachnoid villi. And what they're going to do is they're going to drain that cerebral spinal fluid back into the venous system. So they'll drain that cerebral spinal fluid back into the venous system, and then that eventually, those veins, again, will lead to the heart and lungs, and then, again, be sent out to pick up oxygen and get rid of the waste products, okay? And as that's cleaned out, and again, the different organs in the body, and it picks up, again, nutrients as well as oxygen, then it will come back eventually to the brain in the form of arteries coming up to the brain and eventually make its way to the choroid plexus, and we start the cycle again, okay? So that's kind of how the flow of cerebral spinal fluid goes. This is just showing you those little, again, arachnoid villi that are going to be doing that reabsorption of that cerebral spinal fluid, getting rid of all those waste products, okay? And the reabsorption rate is about 20 mils per hour. And that's going to be equal to the production rate. So the rate of production at the choroid plexus is going to equal the rate of reabsorption at these villi, at the arachnoid villi. So what happens when we, for some reason, aren't getting the proper drainage at the arachnoid villi of the cerebral spinal fluid? And this happens oftentimes, it'll happen sometimes in injury, or sometimes, again, like a traumatic brain injury, sometimes in, meningitis, where you're getting a buildup again of, unfortunately, bacteria and things like that in the cerebrospinal fluid. But it can also happen with tumors as well as developmental malformation. And so this can cause what's called hydrocephalus. And especially you can see that this can be very detrimental in terms of if it occurs during development, because what ends up happening is the brain is really developing in an infant at a rapid rate. And if you end up having this hydrocephalus situation, then the brain actually develops kind of abnormally in this way. And so this actually, unfortunately, has happened to some friends of ours. And unfortunately, because there are things that they could do to actually, save their child's life and be able to allow their child to continue, living. And one of the things that they were able to do was to put in these shunts to where they can drain the CSF, because the proper drainage wasn't occurring through those arachnoid villi. And what ended up happening is this case here, where you end up with these really large ventricles, and the brain and the skull, because the skull hasn't fused yet, the brain is actually, it ends up being enlarged like this, and the skull actually can be enlarged as well. And unfortunately, though, the brain tissue is squished, as you can see, up against here, because again, those ventricles are so large. So unfortunately, the family that we know, these shunts do tend to sometimes block or don't function properly, and so they have to go back in and have the shunts put back in from time to time. But they are able to do this. They are able to put shunts in and drain that cerebrospinal fluid to the veins of the neck and the abdomen. So kind of interesting on some of the techniques that we have today to be able to minimize the effects of this. Blood supply to our brain. So again, blood supply to our brain is really important, because we need that oxygen and we need that glucose for those neurons to function properly. And our brain is something that we can't live without, right? We need our brain to function at that higher rate of functioning. So the arterial blood supply that we have, and we'll get to this kind of in 202, but the arterial blood supply comes to these branches from what is called the circle of Willis on the base of the brain. And again, that's going to supply the majority of our brain with the blood that it needs. Now, our brain is going to use about 20%, even though it's only about like 2% of our total body weight, it's going to use about 20% of our body's oxygen and glucose needs. So this is why deprivation of oxygen for even just 4 minutes can actually do permanent injury to our brain. So again, high oxygen demand, high demand for glucose. That blood-brain barrier, again, is going to help to protect our brain from, again, things like toxins and pathogens, because again, we want to protect that very important organ, right? So proteins and antibiotics really have a tough time passing through that blood-brain barrier. Remember, that blood-brain barrier is, again, going to be created both by the astrocytes as well as the ependymal cells. Now, remember that those cells themselves, again, have plasma membranes. So things that are large and charged, like proteins, are going to have a really tough time. Same with antibiotics. They're going to have a really tough time getting through there. This is why, again, any type of infection that you might have around the brain is really challenging to treat. However, there are other things that are lipophilic, meaning that are able to move right through that membrane, that lipid bilayer, that can pass through. And these are things like alcohol and anesthetics. So this is, again, why alcohol, again, works so well, right? Is that it actually can cross that blood-brain barrier. And that's why you can feel those, again, effects on the central nervous system of the alcohol, because it can do that. Now, again, we have these tight junctions that are going to seal together these epithelial cells that are protecting the blood-brain barrier. And again, we have things like astrocytes and the ependymal cells that are doing that to form that blood-brain barrier. There is some new research that says that there is a lymphatic network that also serves the brain. So we'll kind of get into that more into 202 when we get into the lymphatic system, but kind of interesting stuff. So real quick, let's get into some of the basics on the brain, and then I'll stop this lecture and then we'll get into the nitty-gritties. So remember that the brain, it is one of the larger organs of the body, but it's definitely by far not the largest. But it does weigh almost 3 pounds. Some of the principal parts of the brain, again, we already talked about, are the cerebrum. You can see here again, that's this stuff that's all convoluted, right? And then we're going to have, oops, I'm sorry. Let me go back here. Then we have that diencephalon here, which is going to be in the center here. And the diencephalon is going to have the thalamus, which is kind of our relay station. The hypothalamus, which you'll talk a lot about in 202 when you get to the endocrine system, as well as the epithalamus. And then you're going to have, oh, here's some of the epithalamus over here as well. And then you're going to have the brainstem. So the brainstem is going to consist of the midbrain here, the pons, and the medulla oblongata. And so we talk about why the brainstem is important coming up here and things that it does differently. In terms of acting, again, also as a relay station, as well as some functions that occur in just these areas of the brainstem. And then we'll talk about here the medulla oblongata here, then is going to carry on into the spinal cord. And then you're going to have the cerebellum, which is going to hang back here in the most posterior portion here, kind of posterior and inferior portion of the brain. So we'll get into all of those portions of the brain and what they do. Now we look at the cerebral hemispheres of the brain, okay? They're going to form the most superior part of the brain. So I like to think of kind of the cerebrum as kind of the mushroom here, the mushroom cap here. And that mushroom cap is going to, again, be the cerebrum that covers kind of the top of its stalk, which is if you picture the top of the stalk up here that's covered, would be like the diencephalon and then the top of the brainstem. And then here you'd be seeing the spinal cord coming down here. This is kind of a good way to like picture it. The surface markings that you see, we're going to call the gyri or gyrus individually, are going to be these portions that kind of stick up. And then the sulci are going to be these little kind of tiny valleys in between. So the mountains are going to be the gyri, and then the little valleys in between are going to be the sulci or sulcus, would be the singular. And then the fissures are going to be these really deep grooves that you're going to see, like you're going to see here. So here's a longitudinal fissure, right? That's going to separate the left and the right cerebral hemisphere from each other. And then you also have the transverse cerebral hemisphere, or transverse cerebral fissure. That transverse cerebral fissure is right here, and it's going to separate the cerebrum from the cerebellum. Okay, so you can see a lot of those different major fissures of the brain. Well, not a lot, because you don't have too many to know, right? So pretty easy. And then we're also going to talk about the fact that you can break up the brain then. And again, we do this for our purposes to better understand the brain. So we can talk about the fact that the brain can be broken up into these different lobes. And we break them up into these different lobes to help us really try to catalog the different functions and different areas of the brain. So you're going to see that we can break up the brain into a frontal lobe, a parietal lobe, the temporal lobe, and the occipital lobe, which the nice part is we name these after, again, the bones of the skull that lie on top of them. Okay. Now, there are some different sulci. Again, sulci or sulcus is the singular form of it that we have that help us, again, divide these hemispheres into the different lobes that we see. So here you can see the frontal lobe. If you wonder how the frontal lobe and the parietal lobe are kind of separated from each other, we have something called the central sulcus. So it's this kind of defining sulcus that runs across here that separates our frontal lobe. So this is anterior frontal lobe from the parietal lobe. We also have the parieto-occipital sulcus that's going to separate the parietal lobe from the occipital lobe. Okay. We'll have this fissure called the lateral fissure that separates the temporal lobe from the frontal and the parietal lobe. So again, those different lobes kind of can be separated through these different sulci. So when we look at these different lobes in the brain, we've got the frontal, the parietal, the temporal, the occipital lobes, and then we have the insula. The insular lobe is going to be buried, okay, under these portions of the brain. So you kind of have to pull them back. I think I have a diagram here, really, to see the insula. Okay, so if you pull back the frontal, the parietal, and the temporal lobes, then you could see that insular lobe. Okay? So those are the major lobes of the brain. Those are the major. You should know the central sulcus because, and the reason that you're going to want to know the central sulcus is we're going to talk about this precentral gyrus. So now you know that this is a gyrus. It's the precentral because it's before the central sulcus, right? Since this is the frontal lobe. This is going to have some very specific functions, as well as then this post-central gyrus, which is going to be right behind that central sulcus. So make sure that you know, again, those two terms. They're going to be important. And again, then that parieto-occipital sulcus, you can see here, separating that parietal lobe from the occipital lobe. And again, our transverse cerebral fissure, separating the cerebrum from the cerebellum. So all kinds of good stuff there. And then that lateral fissure here, again, separating up that temporal lobe. So all kinds of good stuff there. Let's see. So again, this is just going through the central sulcus. Again, that separates the precentral gyrus from the postcentral gyrus, the parieto-occipital sulcus, and then that lateral sulcus. Sorry, and you will hear this you'll hear this used lateral fissure or lateral sulcus. It just kind of depends on the book. So you can use either lateral sulcus or lateral fissure. I'll try to stay with lateral sulcus since that seems to be the one that your book is using. Okay, and then each hemisphere is going to have 3 basic regions. So when we talk about, again, these different cerebral hemispheres, when we're talking about the cerebrum, we're going to talk about the cerebral cortex, again, being this gray matter that's most superficial, the white matter here, and then what happens in these basal nuclei that are deep. So it's hard to kind of picture, again, we're breaking the brain up into these different lobes, but then we also can break it up as we go from superficial to deep in each of those lobes. So again, hard to think of the brain in this 3D manner that we do. Now, when we talk about the cerebrum, the cerebral cortex is, again, going to contain kind of our executive suite functions of the brain. This is going to be our site for our conscious mind, our awareness, our sensory perception of the world, our voluntary motor initiation. So what types of voluntary reactions are we going to initiate based on that sensory information that's coming in? It's going to be involved with communication, memory storage, understanding. Again, all of those things that, again, we kind of associate with who we are is going to happen in the cerebral cortex. And that cerebral cortex, again, because it's the cortex, so again, if you remember, that cortex is that gray matter on the outside here. This is going to be composed of our neurons, cell bodies, our dendrites, and those glial cells, right, that we need to support those neurons, as well as blood vessels, but no axons, okay? Those axon fiber tracks are going to be running here through that white matter, okay? And this is going to make up about 40% of our mass of our brain, okay? So the cerebral cortex is really kind of where this is going to be kind of our association integration area for the cerebrum. And when we look at some certain functional imaging, like PET scans and MRI of the brain, that they can show very specific motor and sensory functions are located in very very specific areas that we call domains. And so we can associate these certain areas of the brain with certain sensory functions or certain motor functions, maybe processing certain sensory functions, or again, processing certain motor functions. And so that's how, if you wonder as we're going through the brain, like, how did we, come up with this area of the brain is responsible for, again, initiating a lot of your somatic motor functions. The way that we did this is by using, again, these functional MRIs and PET scans. And so again, and we can see that a lot of our higher functions like memory and language are actually spread over many different areas of the cerebrum and the cerebral cortex. So again, this is kind of cool. You can see this is actually some functional MRI imaging, and functional MRI is just amazing stuff. But you can see, here, this is the area that's focused for hearing, area that's focused again for seeing, areas that are lighting up when you're speaking, and then areas when you're doing these really higher thought processes. You can see there are a lot of areas of the cerebral cortex that are initiated. Okay. So I think I'm going to go ahead and stop here, because then this is really going to get into kind of the nitty-gritties of the next lecture.